먼저 첫 번째 연자이신 닥터 코칸 샴시는 라드MD의 co-founder이자 JN 메디컬 컬리지의 어드전트 프로페서로 계십니다. 1993년부터 지금까지 조영제 및 의료 영상 관련 제약회사 연구들과 FDA, EMEA 규제기관의 조영제 가이드라인 개발을 하셨습니다. 약 25년 이상의 의료 영상 임상시험에 참여하신 분이시며 발표 주제는 라드MD 회사의 소개와 Imaging in Clinical Trials Risk Management Strategies Focusing on Cell Therapy입니다. Now, without further ado, we will turn the time over to the first presenter, Dr. Kohkan Shamsi. He is a co-founder of RadMD and an adjunct professor at JN Medical College. Since 1993, he has been involved in various pharmaceutical research, and Dr. Shamsi also participated in developing FDA and EMEA guidelines for contrast agents. He also has more than 25 years of experience in imaging clinical trials. Please welcome Dr. Kohkan Shamsi.
Good afternoon, everyone. My name is Kohkan Shamsi. I'm principal and co-founder of RadMD. I would like to thank TI and CNR for giving us this opportunity to present RadMD to you and what we are doing currently and what are our plans in future and what are our experiences in clinical trial space. So RadMD was founded based on expertise. My partner, Dr. Rick Pat, is also a radiologist. We both are body radiologists, and we have been working in clinical trial space for more than 25 years. And our objective was to create a company that is expertise-based, because drugs get approved on expertise of the radiologist or other clinicians. Not only we have expertise at the top, but all our leadership team, as you can see, our head of operations, our CEO, our compliance officer, our chief commercial officer, they all have decades or more than 20 years of experience. So what do? We perform core lab services that includes collecting images, QCing the images, developing ECRF, and showing it to the readers. We have participated in over 600 clinical trials. We use best-in-class technology, and we currently have more than 40 employees. Reader services is our core competency. We have more than 300 expert radiologists that are GCP trained. We are #1 provider of expert readers in the industry. At any given time, our readers are participating in more than 200 clinical trials. We also provide consulting that includes regulatory strategy for companies who want to enter European or US market. We provide regulatory strategy. We have experience with more than 42 global agencies. We customize imaging endpoints and modify criteria and develop strategies to proof of concept studies. Our head office is outside Philadelphia. We have offices in London and Miami. Since last 2 years, we are a subsidiary of Medica Group, that is a UK-based company that is one of the largest teleradiology company in the UK. We have participated in more than 600 oncology trials, and you can see all tumor types are listed here. We also have non-oncology experience in terms of diagnostic contrast agent, cardiovascular, Alzheimer's disease, etc. So many companies are involved in early oncology trials. If you go on clinicaltrials.gov, you'll see that 60 to 70% trials are early phase. So early phase requires special attention because they do not have, companies do not have sufficient long period of time like doing phase 3. So we removed unnecessary late phase processes, still being compliant with the FDA regulations, but reducing the workload and customizing the processes for early phase assessment, because early phase studies are done for internal decision-making process, but not for regulatory decision-making. Our consulting services include developing imaging strategies for clinical trials, early development, proof of concept in translational medicine. We also perform late phase studies from phase 2 to phase 4. And for that, we provide regulatory support and also develop risk management strategies. We have, as I said earlier, over 600 clinical trial experience. We have performed image analysis, review of 40 phase 2 and phase 3 trials to utilize that were utilized to gain FDA approval. Just to give you an example, over the last 10 years or so, 18 lung cancer drugs have been approved. 11 of those drugs were read by our readers. I already talked about this expert reader sourcing. We are the market leader in providing reader services to pharma companies, but not only to pharma, but also to all major CROs. Our readers are agnostic to imaging core labs. They perform reads in most of the core labs that are our competition. These readers are exclusive to RadMD, so we can manage their time. And that means that we can predict the read time. And that is very important when the clinical trial is finished and reading is ongoing. Everyone wants to finish clinical trial as soon as possible. Our readers are located in Europe, Asia, Latin America, and in the US. And that include radiologist, cardiologist, oncologist, pathologist, and endocrinologist. We also have a training platform, BRITI, Blinded Reader and Investigator Training Institute. It is the first and global, we are the first and global leader in education and training on medical imaging in clinical trials. We have general courses like imaging in clinical trials or GCP related to imaging, but we also have specific courses like RESIST criteria or Lugano criteria, etc. We are fully regulatory compliant. We were audited by FDA in 2021. And we did not have any findings. So we did not receive any corrective actions notices. Inspector commented that only once before in 18 years has she had no findings inspection. So how do we differ from other core labs? We focus on the independent review results, that is expertise. Conventional core labs focus on imaging data management. We utilize qualified new technology platforms to improve efficiency, cost, and results. Big core lab, if you have had that experience, are limited to internal legacy platforms that were built in 1990s. We encourage customization based on drug mechanism of action and disease. Conventional core labs provide templated reads. Our focus is on early phase, but we perform all phases. Conventional core labs are focused on later phases because they are looking for 600 patient trials, and early phase trials are about 50 to 100 patients. We provide real-time reads. Reports are sent to the clinical team, and we perform collaborative WebEx or to review images. Conventional core labs have standard read platforms. They have timelines for preparation of the platform, preparation of the document like charter and transfer data, and they do it in a very standardized way, and they do not have that flexibility. Thank you. So that was my first part of the presentation. Now I will present imaging in clinical trials. What are the risks of using images in clinical trials, and what are the strategies that we can utilize to reduce that risk? And we'll focus on self-therapy today. So some definitions. The International Organization of Standardization defines the risk management process in 4-step model. So first we need to establish context, and then we need to assess the risk in terms. First we have to identify the risk, and then have analysis of the risk, and then evaluate the risk, and then treat the risk. But we don't stop there. We have to monitor and continuously review the outcome of risk management. What is cell therapy? Cell therapy is placing new healthy cells into the body to replace diseased or damaged ones, or to modulate the function of the patient cells through expression of factors or direct interaction, or the removal of disease-causing or dysfunctional cell using immune cells. A subgroup is CAR T cell products, that are human gene therapy products in which the cell's specificity is genetically modified to enable recognition of desired target antigen for therapeutic purposes. We'll come back to cell therapy specific risks later on. What are the risks related to use of imaging? So the first risk is the trial does not show imaging-based efficacy of drug or cell therapy. So this could be due to genuine lack of efficacy, or wrong selection of cell type, or wrong selection of target, or wrong patient population has been included in the trial. So we'll talk a little bit later about imaging-based eligibility criteria are not met. Then there could be trial design issues. For example, selection of wrong efficacy criteria. I'll show you a couple of examples of that. Regulatory issues, that lack of understanding of risk of utilizing image-based efficacy parameters. General concept in public is that a CT is a CT is a CT. So why should I train the site? Why should I worry about it? And it is not a rocket science. Everyone does. It is a standard of care. But a standard of care in Seoul may be completely different how CTs are performed in Frankfurt, for example. So in a multi-center trial, it plays a significant role. If we have to take that into consideration, that there are variabilities between machines and in the sites. So coming to the first point, risk identification. So I have divided it into 3 categories: planning phase, implementation phase, and evaluation phase. So that is for us utilizing imaging. It is independent evaluation of blinded reads. So we'll go some in more details. But cell therapies have similar risk using imaging in clinical trials, but in addition, they require rigorous toxicity monitoring, and there are some issues in the supply and manufacturing of cell therapy products. So first point was that selection of evaluation criteria that does not or cannot measure drug response. So cancer therapy mechanism, including immunotherapies and cell therapies, are different. Some drugs cause apoptosis, some drugs cause necrosis, and some are anti-angiogenesis drugs. And tumor response basically depends on the therapy. So for example, RECIST criteria, which is commonly used in oncology trial, is based on size measurements. But some therapies cause necrosis without reduction of the tumor size. In these cases, size criteria will not show efficacy. So to give you an example here, on your left side, you see baseline image and a large tumor in the liver. And on day 5, you see significant darker areas in the tumor that represent necrosis. So actually, drug is causing necrosis or destruction of the tumor, but there is no change in diameter. So if you use size-specific criteria here, it will not show the outcome that you're seeing on the image. Another example is acute flare response or pseudo-progression, where you, on the left side, you have baseline, on the middle, you actually see increased size of the tumor, which is a pseudo-progression or acute flare response due to inflammation that is usually caused by immunotherapy drugs. And that size is stabilized in the on the image on the right side. So how do we mitigate this risk? So we must understand tumor behavior. We must understand drug and cell therapy mechanism, how they are attacking the tumor cells. How do we do that? We have to review feasibility data or early clinical data or animal data, both quantitatively and qualitatively, and then select appropriate criteria. And our recommendation is that modify criteria when necessary. People say that FDA or EMA do not allow modification of these criteria, which is not correct. We have done multiple modification many times, and drugs have got accepted by the FDA. The other question comes that imaging modality selected is not appropriate to measure tumor response. So the question is whether if you are using CT or MRI or PET, people just go to conferences and say that that drug trial was presented and they were using CT, so we should also use CT. I think that is a wrong approach. Basically, we need to match imaging modality with the type of tumor and type of therapy. We are still in planning phase. So one thing that I want to emphasize that imaging parameters. Like I talked earlier, a CT is a CT is a CT. When people say, oh, CT is a standard of care, so I do not need to describe imaging parameters in the protocol. So if you have a phase 3 trial, oncology trial, where there are 50 hospitals are involved in conducting that study, each site will have different imaging parameters. And then that creates so much noise in the system that is difficult to assess. Also, we need to define whether you are doing contrast enhanced CT or contrast enhanced MRI. You are doing dynamic phase. If you are doing dynamic phase, for example, in the liver tumors, you need to define exact specification and also contrast agent details. This is an example. On your left side, CT was acquired in hepatic arterial porto arterial phase, and you can see a very hypervascular tumor that is identified by yellow arrow. And on the right side, the same patient, the acquisition is in portovenous phase. So hypervascular tumor, they just enhance, and then the enhancement is gone within 30-40 seconds. So if you were to evaluate the image on the right side, you would say there is no tumor, and you may call it complete response. Although in arterial phase, there is a large tumor existing. So this is the impact of defining imaging protocol in the imaging manual and train the site for imaging assessment. So our recommendation is to prospectively review imaging methodology of feasibility studies, determine best imaging modality and parameters that would optimally measure the response. And you will never be able to completely match imaging parameters from site to site, but try to provide ranges that will provide similar image quality across the sites. We have to learn from early phase imaging that what imaging parameters will work best in phase 2 and phase 3 studies. And we need to train the sites, even if it is a standard of care. So one thing that people do when they start looking for the site, they basically ask, is principal investigator interested in the trial? If he or she has resources, or if he or she can recruit patients. Imaging capabilities of the site are rarely taken into considerations. Radiologists are not sub-investigators. They do not sign any regulatory document. This 1572 is a FDA document that is signed by co-investigators. And many times they are not paid for radiological evaluations in clinical trial. Again, that perception of a standard of care plays in this deficiency. Radiologists and sites are not trained on clinical trial requirement. Imaging criteria used in clinical trials are not used in clinical practice. So I want to emphasize this point, that clinical trial criteria like RESIST or Lugano or Chason criteria are not used in clinical reporting. So if you look up any clinical report, you will not see target lesion and non-target lesion, you will just see a report. So these people are not trained in doing that. So it is important to train them on these criteria. So just to give you an example, if we do not provide these parameters and do not harmonize the imaging, there is an increased risk of getting uninterpretable data. So there have been 10 to 15% bad quality data in recent submissions. So what is bad quality? The reader comes in, independent reader comes in and says, I cannot evaluate images. So images are uninterpretable for that patient. So what does that mean? That that patient is out of the evaluation. So just to give you an example, in 500 patient colorectal trial, blinded readers ascertain that 10% of images are uninterpretable. So that means the data of 50 patients is excluded from the study. This has a huge impact on sample size and statistical calculation. Trial may fail due to lack of efficacy of the drug, not due to the drug is not working, but because the imaging data was not interpretable. So it is really important to prospectively define these parameters. So what we do is we prospectively include imaging in site feasibility questionnaire, because these clinicians hate to fill out multiple forms. So if you give them one form, the compliance is much better. And that gets site radiologists and technologists involved in data acquisition and transfer. The cost of these steps is significantly less than cost of losing 10% patient data or potentially losing the trial. And also, if you consider per patient cost for an oncology trial, is about ranging from $50,000 to $80,000 per patient. So these small steps that you can take are significantly cheaper if you do them prospectively than to lose the patient or to lose the trial. So coming to implementation phase, in the study planning, we need to plan prospectively that blinded reading and or site read will be performed, because many times when they do early phase trial, they'll say we will only do site reads. But then FDA comes back and says, no, you need to do independent read, or they hear from someone else that competition is doing independent read. So they come back and say, now we want to do independent read. What happens that you have not planned, the cost is increasing. And then they say, oh, it's too expensive. And also not only cost, but also time. You know, independent reads usually are delayed and they are performed after last patient has been recruited. So if you do not plan that 3 or 4 months delay, there's a lot of frustration that comes in. Regulatory agencies accept blinded read results over on-site evaluation for proof of efficacy. And then there are well-established differences between the site versus central read. Blinded reads tend to show lower response rate. That is natural because the site readers have patient information, lab information, and all other data. So site evaluation of efficacy data has a lot of site variability, because if you have 50 sites, there are 50 radiologists, at least 50 radiologists, because they may be other radiologists may be on that duty with when this follow-up time point is being assessed. So let's say 50 sites, 50 radiologists are evaluating who are not trained. So there is a lot of variability in assessment. Confirmation of progression is a big issue, because site may call that patient has progressed, so the patient is taken off the trial. When that patient comes for blinded read, that follow-up time point is not available, because the site has decided that patient has to be taken off from the trial. So that patient is taken off. So for blinded reader, that patient is gone, because there is no follow-up time point. So that phenomena is known as patient censoring. And again, safety reads are also very important. For example, in Alzheimer's disease immunotherapy trials, a phenomena of amyloid-related imaging abnormalities have been reported. And this has increased the cost of Alzheimer's disease trials significantly, because all FDA mandates that all immunotherapy trials you need to follow the patients every 16 weeks or 12 weeks to manage this amyloid-related imaging abnormalities. And then with CAR T cell, significant neurotoxicity has been reported. If you look at the package insert of Chimera, I think it was 61% or 60 to 70% patients reported neurotoxicity. So to summarize, like I said, imaging criteria like RECIST are not used in clinical practice. Therefore, radiologist clinical interpretation is designed for patient management, not for clinical trial. And that does not match with the trial requirements. And clinical reports are generally insufficient for selecting and measuring target lesions like we do in clinical trials. And I talked about that the images are clinically interpreted by multiple site radiologists using non-standardized terminology, multi-site and multinational. And then you have every radiologist, including me and him, write in our reports to please correlate clinically, please follow up with this and that. That is not done in clinical trials. So what we recommend is collect site imaging capabilities prospectively. We also do sometimes review of sample cases before the first patient inclusion. Rigorous site technical training, and we strongly recommend to our sponsors that compensate the site for quality data. Because as I gave you the example, this is very small payment to save that additional cost and pain. And identify radiologists as co-investigator at the site, ask them to sign relevant forms so that they feel responsible for image quality and protocol adherence and image transfer. One thing is that clinical CROs, and we have trained many clinical CROs, site personnel, CRAs and project managers and biostatisticians. They are not trained for imaging issues. When they do site monitoring, they usually do not monitor imaging. They just say that, have you performed this 12 to 14 weeks follow-up time point? Yes or no. Have you submitted the images? Yes or no. But I think it is important to train them, and also training on safety of cell therapies. Because how do you detect neurotoxicity? You need to do MRI, right? So that also involves imaging. So imaging is also performed for eligibility reads, patient eligibility. For example, many trials have image-based inclusion criteria. So number of lesions, size of the lesion. The inclusion criteria is evaluated by PI, who is a non-radiologist most of the time. Oncologist or neurologist will evaluate and say a patient can be included. So in one of the presentations by FDA and NIST is a standardized organization in the US. They said 10 to 15% of the patients were inappropriately included. So this has a real huge impact. And this is not detected until blinded read is performed. So for example, you need to have less than 2 centimeter lesion, or at least one lesion has to be more than 2 centimeters. And the blinded reader comes in and says, there is no more than 2 centimeter lesion in this patient. So there is no RECIST criteria then. So it is only detected, but by that time patient is gone. And so this can have huge impact because patient enrolled in a study based on incorrect interpretation can result in statistical issues because those are thrown out of the trial for assessment, could be unethical to enroll patients that are not qualifying to be included. And in the US, I always put legal issues because during the blinded read, these are considered as dropouts that could result in negative impact on drug response. So how do we mitigate this risk? So we recommend to do independent read of eligibility. So the baseline images are acquired by the site and sent for evaluation to the central image management facility. Images are evaluated by independent radiologists to verify image-based enrollment criteria. Site is notified for inclusion, whether the patient is included or not, includable or not. This leads to decrease in percentage of patients inappropriately enrolled in the trial. And our turnaround time is about 3 to 5 days. This is an example where the criteria said that diameter should be less than 4 centimeters, and this patient was included with more than 10 centimeters tumor. So just to give you an example, in phase 3 oncology study for metastatic hepatoma, 400 patients were enrolled. Enrollment required 3 measurable liver lesions of more than 2 centimeters each. Patients selected for inclusion by clinical CT reports reviewed by site coordinator, not even by a clinician, but site coordinator. At blinded read, 60 patients did not meet enrollment criteria. Benign lesions like focal nodular hyperplasia, adenomas were included. So a study was reopened for additional patient accrual. Submission was delayed for additional 1.5 years. So that is significant cost increase, and patients were inappropriately treated. Like benign lesions were inappropriately treated. So coming to the last part, independent evaluation, reader selection, reader performance, adjudication rates are all risks that if you do not mitigate prospectively can impact the trial. So what are acceptable adjudication rate in oncology trials? So what is adjudication? When reader 1 is saying that there is progression, when reader 2 is saying is stable disease, then the third reader comes in and adjudicate whether reader A is correct or reader B is correct. So in Oncology Drug Advisory Committee, they flagged one prostate cancer trial that they had 39% adjudication rate, and ovarian cancer trial had 41%, and lymphoma has 51%. So they basically said that if your readers are not agreeing 40 to 50% of the time, how do we know that your drug is working or not? So we use multifaceted approach to reduce high adjudication rates. We begin with imaging endpoint selection and evaluation criteria. Then we select readers who are similarly qualified and have similar experience in clinical trials. We perform standardized reader training. There's a pre-read training, then read-specific training, and testing by expert radiologists. And we basically criteria also have a lot of ambiguous areas. For example, how to deal with hilar lymph node, how to deal with pleural effusion or ascites. So we create a reader agreement document that whatever you do in clinical practice that's different for this trial, readers have to agree that how you will deal with this. We are not telling them how to deal. They talk among each other and then say, we are going to do this. and we monitor their performance by that reader agreement document. So how to mitigate this? We, like I said, performed by controlled pool of radiologists with a specific experience. We train and test. And we do ongoing reader performance monitoring. So initially, when they have read 10 patients, we go back and look whether they are following that reader agreement document or not. Some word about CAR T cell safety monitoring, because that is specific to cell therapy. About 70% of the patients showed cytokine release syndrome and then neurotoxicity. So on your left, A is showing encephalitis-like features, B is a stroke, C is leptomeningeal disease, and D is vasogenic edema. So these things were seen in recent CAR T cell trials. So it has become important now that how to monitor these and what will be the frequency of monitoring these therapies. So we will have more and more imaging involved in safety monitoring of these cell therapies. So we should be prepared and be trained internally, as well as we should be training the sites for these monitoring, because these are new entities that were not seen before. So I was asked to give a couple of things about regulatory guidance, and this is probably my, am I okay on time? So as a class of therapeutic agents, cell therapies include stem cell-based products, and this is from FDA guidance document, that meet the definition of following regulations. So they come under biological products, they come under drug products, they also can be device products, xenotransplantation products, and human cell tissue and cellular and tissue-based products. So this is basically taken from FDA, articles containing or consisting of human cells or tissues that are intended for implantation, transplantation, infusion, or transfer into human recipient. And these are the FDA guidance reference there. There is a specific mention of guidance for CAR T cells. We recognize, and this is from FDA, we recognize that development, manufacture, testing, and clinical assessment of CAR T cells is challenging. Careful design and appropriate testing of CAR transgene and delivery vector are critical to product safety, specificity, and function. So that is again an important point, because manufacturing of these product also has limited lifespan. So in site selection process, you need to have that kind of thing in mind, that how far the facility is, or because the efficacy goes down if it is kept for a long period of time. So to conclude, plan early, selection criteria appropriately, select imaging modalities, readers, site, have early regulatory input. So don't be afraid to go to FDA and ask questions. A lot of people say, I don't want to go to FDA, but we recommend to prospectively go to FDA or EMA and ask questions. Develop reader profiles, that what kind of readers you would like to have. Because many times people require experienced readers, but now with